Okay, my friends, exciting new news today. We're going to start a new format. Daily, I will do the news. I'll call it MU News, and then a date, and then maybe a topic name. Today, we're going to be talking about Alzheimer's, because they just approved a drug to treat Alzheimer's. Now, let me just discuss, because these are going to be very, very, very deep. I don't just get to the surface. I go right to the bottom. And they're going to talk about what the drug is, what it supposedly does. They're going to talk about this buildup of Lewy bodies and plaques and so forth that accumulate, and all of the nerve synapses, and how the nervous pulses work, and how the neurons are constructed. I'm going to go right to the end. And the end is going to be the blood-brain barrier and ribosomes. Remember those two terms. Okay, my friends, this has been in the news extensively, and Parkinson's and all the other diseases, because there is a common thread which I will discuss shortly. The FDA approved this Alzheimer's drug for early disease symptoms. Now, we're going to go deep into what the symptoms are and why they become problems. And this is all about, here's another one 5 days ago, signal of future Alzheimer's may be hidden in the way you speak. Alzheimer risk strongly shaped by your mother's side, study finds a couple days ago. I mean, it's all about Alzheimer's, Parkinson's, autisms, neurological diseases. And what is a neurological disease? What is that? It's where your nerves either die or they don't communicate. All right, let's just start from there. All right, here's what they have never been thinking about, I don't think. We're talking about the blood-brain barrier. So somewhere in your neck area, there's a barrier that says all your big things stay down below. The brain, we can't contaminate. So it's a complex regulatory interface. It prevents unregulated leakage of serum proteins. Proteins are big into the central nervous system because they're just too big. So it allows only select molecules to pass from the bloodstream into the fluid that bathes the brain, all right, such as vital nutrients, while blocking toxins and pathogens, which are big molecules. The blood-brain barrier is highly permeable to oxygen, little tiny molecule, CO2, another tiny molecule, water, H2O, tiny molecule, and most fatty soluble materials. If they're fatty and soluble and they can dissolve in water, they'll go through there. Like alcohol will go through, and anesthetics. It is also slightly permeable to plasma electrolytes, but impermeable to plasma proteins and many other materials. You can't get those things up through there. So how do you get those things, which are enzymes, up into the brain? You have to get those enzymes up there, and they're huge. I can tell you exactly how it's done. Okay, I am going to be discussing primarily enzymes, but you do not get an enzyme without a bacteria. The bacteria create the ribosome creates an enzyme. And they're finding out that bacterial enzyme traps and breaks down these forever chemicals. The enzyme comes and closes around the troublesome chemicals. They're trapped inside and they're broken down. It's exactly what these things do. That's what enzymes are designed to do, and they do it instantaneously. Very, very quick. You see this? These are the forever chemicals. PFS chemicals, a group of over 10,000 man-made chemicals, long carbon fluorine bonds, make them very stable and persistent. But the enzymes can break these things down. Enzymes are very, very exotic. All right, look at this. This is amazing. You see these different colored areas? They indicate different chemical compositions. This is the open state of the nutcracker. It gets something in here that it wants to crush, and it crushes it. It's an illustration of the nutcracker enzyme in its closed A state and its open B state. The blue part of the enzyme, and then it goes on to talk about it. But wait till you see what an enzyme looks like. Oh, watch this. You see that? That's an enzyme. You see how complex that is? It's like, it's all magnetism. They're all magnetic particles, just like this. Virtually almost identical to like this. Now, you see these different color regions? It appears that there are regions within enzymes that have specific functions, like that nutcracker cruncher. All right, it sure looks like the same thing to me. You know, whether those colors are real or not, I don't know. But this is about a year ago. They're trying to understand enzymes, finally, thank God. Because they do everything. Enzymes do everything. And without bacteria, you got no enzymes. All right, here it is right here. Bacteria are the factories that produce enzymes. And I'm going to show you this biologically. All right, so they are the factories. You don't have that factory, you don't get the product. All right, let's start from the beginning. Where do enzymes come from? They come from the factory, which is the bacteria. And I don't see any enzyme. Where's the enzyme? They're in these balls. All right, these little bitty tiny balls. You see them all over the place in here. This particular bacteria has its own DNA. You see this? This is the DNA strand. Now, that DNA says, I'm going to be a bacteria, and my job is going to be to create a certain exact ribosome that will look exactly like this when it unfurls out of that ball. Because the ball comes out. The ball is just tiny. The ball is like maybe this big. All right, and it's got all that stuff inside of it. And when it opens up, poof, they pop open. I'll explain that in a second. Now, and it has a design that says, I'm this kind of bacteria and I create ribosomes. Other ones are going to say, I consume waste. All right, but normally a ribosome comes out and breaks things down. Then you have other ones that go and consume it and take it down to the lymph nodes and drop it off. And there's all kinds of, they do the cleanup, they feed us, they break down the food, they kill the invaders. They maintain the arteries and veins and all of your muscles and all of that business. And, you know, they do everything. Bacteria is us. And there's more of them in us than there is us in us. And I have a very good friend who is a triple PhD in bacteriology, very, very deep. And he owns his own company. They do big food service bacterial analysis so that, you know, the food supply doesn't get contaminated. You get a whole bunch of labs. Last I knew, he had like 19 labs running in these food processing companies. So anyway, and I'm going to be having a video with him tomorrow, and I'm going to be going over all this stuff with him. And if he has any issues with what I'm saying, he'll bring them up. Now, Andy's a really nice guy, and he's a super smart guy, and he's pretty much endorsed the things that I've been saying, and to be able to engage with him, you know, one-on-one is going to be very good and helpful, because he's a triple PhD. He's the kind of guy they can listen to. So I'd like to get this stuff published somewhere, because this is what we need, these ribosomes. The only way you get the ribosome is to have the enzyme, the bacteria. You don't get the ribosome, you're done. Now remember I talked about the blood-brain barrier? It's a tiny, tiny, tiny little orifice that goes in. Think of it that way. And these balls are tiny, 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 tiny balls. They can go through there. So ribosomes can go through. Proteins and other things that are opened already, like this, that can't get through there. What we're talking about is something down about this size. And that can get through. That can't get through. It's just too big. This size probably, let's say, can get through. And then when it gets through the other side, if it gets called upon. Now, why would it get called upon? Because there's another chemical signature from the lysosome that says, I got to come over here and slice you open, and I'm going to pick on this exact bacteria, ribosome. Out it goes to do its job. All right, we're going to be looking at the anatomy in a minute, but with the lysosomes are organelles found in animal cells, function as cells' digestive systems. They break down and recycle these big molecules that you got to get rid of, such as proteins, carbohydrates, lipids, nucleic acids, both inside and outside the cell. Process is called autophagy for internal components, and endocytosis for external materials. Now, all it means is that lysosomes are the trigger that says you got to break down something. They don't just go breaking things down for nothing. They have to be told how much to break down, what to break down, who to send out to break it down, and that's who they go to the different ribosomes. Where do the ribosomes live? Well, they live in every single cell. All right, I'm going to show you all this stuff, but even the neurons contain ribosomes, those little tiny balls. They're large multi-protein organelles that synthesize proteins in all living cells. Synthesize means to break down and reconstruct. Ribosomes are found in the neuronal cytoplasm, I'll show you in a second, along with other organelles like the nucleus, mitochondria, and Golgi apparatus. All of those things in every single cell in your body, they have to be maintained, and the ribosomes do that, working with the lysosomes. All right, I'm going to make this extremely simple. These are only single cell. These are very small little living organisms inside of you, and they produce the ribosomes. They squirt those out, and the ribosomes, this is what happens with the ribosomes. All right, the ribosomes contain the enzymes. Did I show you that? Let me just show you what happens with a ribosome. This is the ribosome, the little bitty ball, little tiny ball like this. It comes out and then it bumps into something that chemically triggers it. And the trigger opens the sheath like this, and it gets gigantically huge enzyme like that. All kinds of different things going on. Now, enzymes and proteins, they both come out of the same package. One of them is there to build as building blocks, and this one is a breaker. This breaks down food, it breaks, and there's all kinds of different enzymes. Not just one says I'm going to break down all the food. No, there's one that says I do a bunch of different types of fats, and I do some acids, and some carbohydrates, and I do some sugars, and I have an acidic value, I have a caustic value, or they're enzymes. They do an enormous amount of chemistry instantaneously. I call it click chemistry. And it is when they come up against something they don't like, click, and it's gone. It's dead instantaneously. And then they just keep going on. It doesn't even use this up. It goes on to the next one. Click, 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 and it's that fast. Enzymes speed up a chemical reaction, and what they use is half-lives. This is the problem. The people that are working on this stuff, they don't have a full complement of understanding of the biology of what all this stuff is made of. And here's where these ribosomes are. You see the little balls? You see, this is cytoplasm. Every one of these little balls is one of these ribosomes. And this right over here is your lysosomes. And they send out the signal to trip off the enzymes. Pop them open. Ribosomes pop open into enzymes and go out and do their job. And it has a membrane, just like they all have a membrane. It's got a Golgi apparatus. It's a nucleus. It's got all that stuff. And this is a neuron. All right. Now, myelin sheath is another thing. The myelin sheath is an insulator, and that goes bad too. And here's what they're talking about: microtubules, this is a nerve cell, and it's sending nervous signals out of here. They really don't understand it, to be perfectly honest with you, but they know they come out of here, they transfer through to go somewhere, and they're just nothing more than things like this. And every different color is a different protein, and there's a different amino acid. And when they gang up together, they have to go in a certain sequence and to make the signal transfer. So in other words, if you're coming down here and you have a break in the signal, you want to be reattached, boom, you get reattached, you're okay. But here's what happens. Let's say somehow you can't get reattached, and every time you try to get reattached, it just goes away. It won't touch. All right, that's the polarity. I think that might have something to do with something here, but I can tell you one thing. As far as I'm concerned, this is basically what they're talking about in these sequences. Now, if you short through here, through the myelin sheath, you're in trouble. And I think that's what's happening. And maybe these things are all working fine. But first of all, you have to have the ribosomes. And you see how many ribosomes there is in there? I think they must have, and I don't know this for a fact, I'm just guessing, but I think they must have virtually all the ribosomes in your body have migrated to virtually every cell. And they're just waiting around, waiting to do their job if they're called on by the lysosome. The lysosome sends out the signal to open up the ball as a trigger and opens it up, and it goes and does whatever it's supposed to. And these are very, very, very sophisticated. These are like little, almost, if you could think of it this way, these are almost like arteries, and these are like little blood vessels. And then at the very, very, very tip of the ends, they're like the, oh boy, they're like the little lymph nodes at the bottom of the capillaries. All right, and at the capillaries, anyway, your body is designed just fabulously. So my feeling here right now is if you don't have the enzymes to create the bacteria, you have to have bacteria to create the enzymes, you're not going to have the ribosomes. You don't have the ribosomes. I don't care what the lysosome says to do. You don't have the product to do the job. And that's why people have these never-ending diseases, and they just can't get rid of it because they can't get the ribosomes. So the point being is that we need a bacterial database of what everybody's body has for bacteria, and you could get a pretty good estimation just by doing stool sample analysis. And it would create a database of what seems to be healthy versus unhealthy people, and it'll float around, and you'll get to the point where everybody that has these different things is going to be suspect to be unhealthy because of a certain invasion, whatever. And all of that stuff should play right out on a bacterial database, and right now we don't have that. You should be able to go in and say, here's a piece of poop, what's wrong with me? It really could almost be that simple. Okay, so my claim has been for many years that what you need to do is replace the bacteria in your gut. And they found now also Parkinson's, which is another neurological disease, is linked to gut bacteria, suggests an unexpected simple treatment. The gut bacteria is bad in your gut. You know what the treatment is? B vitamins. B vitamins are, I always consider them just to be minerals. They're nothing to think too much about. But it appears they're co-enzymes. Like B12 is extremely exotic. It's as exotic almost as an enzyme. And I showed you what an enzyme is. That's an enzyme. Now B12 has a core metal. I think it's cobalt. I think so. And there's a whole batch of surrounding particles that are what they call ligands that attach to it. Now, a coenzyme means this has to do a job. But sometimes you need, like a car, you need gas, you need oil. Well, gas and oil, that type of thing. Just think of it that way. You need salad dressing, maybe some tomatoes, little croutons. Anyway, whatever it is, that's the deal. You need these, and they found out these vitamins are co-enzymes, and they also make this reaction happen much, much faster than it would, even just with the enzyme. And enzymes make things go just All right, listen to this. Enzymes are complex chemicals produced by bacteria, the things that we're talking about. And they come out as ribosomes. So they're complex chemicals produced by bacteria, speed up chemical reactions without being used themselves. They're made up of amino acid subunits, which I think I showed you or will. And they are not alive. They're not alive, these enzymes. They can't reproduce. And or consume waste. All right, the enzymes themselves can't consume waste, but the enzymes are involved in many bacterial processes. Now, the bacteria, though, do consume the waste. They come around and eat up. They do what they call apoptosis, where they eat up the dead cells and everything, because you all got all kinds of things in your body floating around as trash, and you got dying body parts continuously, thousands and millions a second probably, and they got to be consumed and flushed out. So that's what the bacteria does. Now the enzymes break them down to where they can be consumed. Now, these enzymes are involved in cell wall formation. Enzymes like transglycerases. Anytime you see an ACE on it's an enzyme. Transglycerases, transpeptidases, carbon peptidases, and endopeptidases incorporate new peptoglycan into existing peptoglycan to form a rigid cell wall. That just means the cell walls like this, you can add more and more and more. So the transpeptidase is what transfers it into the rigid cell wall. That's what all these things require, enzymes. DNA hydrolysis, this restriction endonucleases, another ACE, cleave phosphodiester bonds in the DNA backbone. Phosphodiester has something to do with phosphorus, which has something to do with the membranes. They're phospholipid membranes. Every single cell in your body has that. So it cleaves that membrane. And let's get into the DNA background. That's another enzyme. Urea hydrolysis, urease, another ACE, hydrolyzes orally administered urea to carbon dioxide and ammonia. Whatever that has to do, I'm not sure. Proteolysis, enzymes break down proteins into their component amino acids. So you eat something that's got a protein. It's a pretty complex molecule. Well, these enzymes come up and go, done. And it turns in, it's just instantaneously. It's absolutely stunning how fast they work. All right, this is how fast enzymes do things. How many reactions do enzymes carry out each second? And this is from very detailed sources, and they reference all of them. You see them down here. Now here's what it's saying here. The biochemical reactions taking place in the cells, though thermodynamically favorable, are in most cases very slow. They're just normal, just sitting around. If left uncatalyzed, that means there's no presence of an enzyme. They're just sitting around doing nothing until an enzyme comes in to do a job. Listen to how fast they work. Now, for example, the spontaneous cleavage, that means cutting instantly, of a peptide bond, which is these DNA bonds, would take 400 years at room temperature. 400 years. And phospho-non-ester hydrolysis is just what we were just talking about, routinely breaking up ATP to release energy, which you have to do all time, all day long, would take about 1,000,000 years. 1,000,000 years in the absence of the enzyme that shuttle that reaction along. And this is the paper. Fortunately, metabolism is carried out by enzymes. These are these really fancy little enzymes that often increase rates by an astonishing 10 orders of a magnitude or more. There's a paper on that. Anyway, it's talking about what an enzyme does, how fast it works. 400 years at room temperature. This would take 1,000,000 years, and you're needing this all day long, instantaneously. You can't live without it. This is serious. These are what enzymes do. And the only way you get the enzyme is a bacteria. Said it a million times. Okay, AI wrong again. These are ribosomes, these little tiny balls. They exist in the cytoplasm. They're not huge molecules, which AI claims they are. When they open up and unfurl, yes, they turn into gigantic enzymes, but they can only get up here because they can get past the blood-brain barrier, and they are produced by bacteria. And these are the actual nerve fibers, microtubules, and they look to me like they're showing them as little dipoles that send a signal down, back or forth. But these are all the ribosomes, and this is all cytoplasm, all that fluid, and they exist in there. And when they get called on by the lysosome is the cracker. Cracks them open, and they pop open. All right, see, they sort of understand it, but sort of don't. Lysosomes function as the digestive system of the cell, I agree, serving both to degrade material taken up from outside the cell and to digest obsolete components of the cell itself. I also disagree. I also agree. But how do they do that? They don't have the components to do that. They call upon the ribosomes, which are just floating around waiting, and then they send out a certain signal to say, hey, we need you to go down and do whatever it is. We just digested some fat, and you need to go down there and break it up or whatever. And that's why there's so many of them sitting around. That's the only thing I can think now, is these are all ribosomes in the cytoplasm. Okay, this is just backwards. HIV infection can cause digestive issues. No, the digestive cause HIV. Gastrointestinal inflammation, diarrhea, nutritional absorption problems, abdominal pain, dysphagia, bowel incontinence, bloating, constipation, nausea, and vomiting. That's all because you're not digesting right, because you don't have the right bacteria. That's my opinion. All right, here's right here. Even long COVID. This is from Harvard. Few people, even in the medical field, are aware long COVID symptom may include chronic diarrhea and abdominal pain. The doctors don't ask too much about your digestion. That's the issue. And you can tell how your digestion is working from fecal samples, but they have to be analyzed, and we need a database. All right, this is a pretty good synopsis. They're talking about membranes and so forth, and it allows the cell to control what comes in and what goes out. The interior of the cell, inside the membrane, contains a number of different organelles, very complex, including mitochondria that produce energy. Hold on, let me just show you the cell, and we'll come back in 1 second. All right, here's what's inside of the neurons. All right, and these are all these little organelles, and Golgi apparatus and membrane. Of course, they all have membranes. Then you have mitochondria. You have a myelin sheath that covers everything. You got microtubules, lysosomes. You got the endoplasmic reticulum and the smooth and the rough, and then all of these dendrites. And what do these do? These connect to other cells. So you have a gigantic mechanism of attached spine cells. So you could create one gigantic organism, and then apparently this either comes out and goes out of each one of these neurons, or they all match together somehow, and one of them is assigned to deliver the stuff. I have no idea. But this is where these microtubules do the connection, and that is called the axon. Here it's, I thought I had something. Hold on. Oh yeah, here it is. All right, well, this is not good. Hold on. It's a pretty deep rabbit hole, but it's not hard to understand. Glial cells are non-neuronal cells. That means they don't create signals, they're structure. They're in the central nervous system, the brain, the spinal cord, basically everywhere, for a peripheral nervous system. They're the conduits. They do not produce electrical impulses. All right, but these are the glial cells. All right, these are the neuronal cells, and they send out their signals, and they seem to attach to other cells in a number of different ways. And this is what it says here. The image shows the 4 different types of glial cells found in the central nervous system. 4 different types. Epineal cells, which is the light pink. All right, see, this is the surface, membrane, basically. So they call them epineal cells. Now, astrocytes are the green cells. That's these astrocytes. Microglial cells are the red cells. These are microglial cells, or maybe these. And oligodendrocytes, functionally similar to Schwann cells, those are the protective ones, are the light blue. The light blue is the sheath, the Schwann cells, and I suspect they have something to do with damaged nervous system, something going wrong. You can't convey the image, you can't talk, you can't, all kinds of things. And this is a lot to do with autism, Parkinson's, Alzheimer's, dementia, being able to recall. You need chemistry. You need the right chemistry. You need to protect this. I'm going to show you something that's going to shock you, I'm sure, but I'm going to show you. Remember how all of these fibers are in brains. I'm going to show you a brain scan, and it's going to show all these different colors because they're different chemistry. And they're actually different colors leading to different places in the body that are, I believe they're going to be muscular or feelings or something. But I'll show you what it is. And there's a fossil that shows it as well, and was preserved in crystal. All right, this is a brain scan of a patient's head, all the different neurological fibers that are going from here to there, and all these different colors indicate a different type of chemistry, a different type of signal. And the blue here is the neuronal mass, and then these are the axons, all that are coming out to travel to go wherever they go, and then it all goes down into the brain stem and so forth. And I'm going to show you something, knock your socks off. You see this ball here in the back? This right here, my friends, is a crystal skull. It was the Mitchell Hedges skull, been dismissed throughout history as being fake. It's not fake. This is the exact same pattern of fibers, identical, no difference. The face was on its like this, so the lip was pushed back, the nose was pushed back, and all the flesh and all that stuff goes away. And what remains was replaced by silicates. And that happened quite a bit. I have silicate replaced things here in my shop, but this just happened to be a skull. Somehow it happened just perfect. And these are, the brain moved forward this way. You see how it bumps back normally? Well, it's because it was on his face. And that's why the lip is twisted up. It's just amazing how body tissues do something called nucleophilic substitution. And that means it's basically nucleosynthesis, which is the nucleus. They resynthesize them and turn it into something new chemistry, which is silicon-based, in this case, entirely, basically. All right, so you just saw how many fibers were in that brain. What's in each fiber? These are the fibers. Now, this is coming down the brain stem or something. All right, but these every little nerve fiber is like this. They come in little bundles, and the whole thing is sheathed. And then in the bundles, there's little semi-bundles. And in the semi-bundles, I believe there's even additional bundles that are in these. But every one of these bundles is insulated, and every one of these fibers is insulated. And if anything goes wrong here, you got a problem. Because these control something just like a wire in a house. If it doesn't work, it doesn't work. This is what it looks like cut this way. This is the reality of the situation. I think I have a better shot of this. Hold on. Yeah, here's one. All right. You see these here? You can see it's just like a wire. It's just like a wire in the middle of the sheathing. All right? And this is the membrane that protects the whole thing. So you got another membrane around here. Every time you see these divisions here, that's a membrane. Then in between you have the cytoplasm, you have the extracellular matrix of fluids going around. And that's where the ribosomes are. They're all in here. All right, they don't show it, but when they say fascially, that's fascia, that's the coating. All right, nerve fiber, myelin. Myelin is that's extremely important, and that is also created by enzymes. So here it is right here. Myelin is a fatty substance. It's a gooey, slurpy substance. It covers nerve axons, which are the tubes, in long sections made up of about 40% water, 60 to 75% lipids, which are fats, and 15 to 25% protein by dry mass. Its main lipid fat is, whatever that is, glycoside, a glycolipid. All right, and other major lipids include cholesterol, ceriboside, various phospholipids. These are the membranes, phospholipids. Myelin also contains proteins, including protolipid protein. Proteins are made by the enzymes. I showed you proteins are created by the ribosomes. You see this? These are the ribosomes. All right, they get squirted out of the bacteria, and then they go and they just seem to be sitting around in every cell. And then something triggers them. The lysosome says, hey, you got to turn on and create whoever he's talking to. There's going to be a bazillion different ribosomes because every bacteria creates a different type. So he's going to communicate the lysosome guy to the right one. The right one is going to unsheath, and then it comes down to this. It's either an enzyme or a protein. Depends on what you need. It's not going to be the same ball that can produce either one of these. It's going to be a different ball. So you're going to have like a ton of different colored balls floating around inside of every cell in the cytoplasm. All right, you see this? Cytoplasm. That's all of this here. And these are all the ribosomes. They're just floating around all by themselves, having a nice time, doing nothing. They got a layer around them. They're not alive. Here's the key. They're not alive. They don't eat anything. They don't do anything. They're just like a little plastic ball. And then somebody comes along from the lysosome guy and says, go get this guy over here, the pink one, and he comes over and he goes, and it pops open, and it opens like this. Or he might come over and drag it out and bring it to somewhere, wherever it's needed. I don't know exactly at that point, but I can tell you one thing, no question whatsoever. These open up from those capsids, the capsule, and pop open into these huge, huge exotic particles, which are these. That's the only way that you can get through the blood-brain barrier is because they're ribosomes. Ribosomes, and they don't do anything. They don't eat anything. They don't do anything. They're just chemistry. It's sitting around waiting to be used. And when they break open, that's when they take on a life of their own. But otherwise, they're just sitting here waiting. The lysosomes are the trigger. Now, who tells the lysosome what to do? I don't know. But the whole thing works together. So this is where it becomes quite mysterious. And of course, there's the myelin sheath. And if you don't have that pristinely perfect, you're going to have all kinds of interruptions with your brain signals. Now, are these people losing their minds and, dementia and all of these different neurological diseases because things are dying? I don't think so. Because I've seen a lot of people come back based on repairing their gut, literally repairing their gut. And they recover from, well, I don't say recover. I never want to make any medical, I give no medical advice. I'm not a doctor. I don't do any of that. I'm just saying from what I have seen, all of the studies, they talk about the gut bacteria causing or having some effect on these issues. I mean, it's right across the board, and I know I've shown it over and over and over and over and over. So there's no hiding it. Everybody understands it now. And these are, this is all about the chemistry and about the neurons and the mitochondria producing the energy. And this is the lysosomes, capable of breaking down proteins, lipids, and carbohydrates. It's not the lysosomes. The lysosomes trigger the enzymes. All right, to survive, cells must take in nutrients from outside the cells and remove the waste that builds up inside the cells. Neurons, more than any other cell in our body, must keep the inside of the neuron different from the outside and control the movement of substances across the membrane. That's the job of ribosomes, to intervene. All right, so they say neurons signal transmission cells. Well, what does that mean? In the previous chapter, we mentioned that nerve fibers emanate from nerve cells. Okay, so the fibers come out of the cells. They're called neurons or brain cells, which are responsible for generating, carrying electric signals, just like a wire, throughout your body. Neurons are present everywhere in the nervous system, which is your entire body. The brain, spinal cord, periphery, nerves, nerve fibers are all made up of axons of the neurons. The neurons are the brainy part, and then the other part is the axons is the fibers. So the neurons have a unique structure that helps them do this example following. All right, let me hold on here. All right, this is the neuron, or we call it a nerve cell. Same thing. Now, these are all the little branches at the end where all these fibers emanate from, through this myelin sheath coated axon. All right, now they show them little chunks, the blue in chunks. Now, I would think that's just so that they can be very extremely mobile. Now, are they that far apart? I don't know. But I can tell you one thing, that thing has to be insulated, no question whatsoever. It has to be insulated from everything that's around it. It cannot be involved in anybody else's signals or anybody else's fields. All right, these, they call them Schwann cells and fascia and, you know, whatever it is, it's a coating, a fiber. Now, at the other end, all of those wires come out and they go to all kinds of places, dendrites, just everywhere. All right, so everything conducts in this way and back that way. Now, I don't know if all of them go both ways or some of them just go that way and some of them come back. I would think they go only one direction myself, but I don't know. All right, I know I started with Alzheimer's, let's see, but Parkinson's is the same thing. It's linked to gut bacteria. Now, you see these little gut bacteria? This is what they are right here. I'm going to go right through the whole process. This is the bacteria, and they are just living little cells that get into you from I don't know where. But you sort of ingest them into your body, and somehow they accumulate it in your digestive system and so forth. And they created the ribosomes. So out of here, they come out and they create these ribosomes, which are the balls. The balls come out and they program the messenger RNA, and it incorporates into our DNA and gives us, you know, immunities and all kinds of things, or it could quick kill you. It goes both ways. So you need to have the good bacteria in you to fight off the bad bacteria. But this is where the ribosomes, which this thing here creates this, and that's that ball, that ribosome. And the way it does is all of these are the program codes that come out in a polypeptide chain. And then they come into the transferase, which transfers these into the messenger RNA inside this cloud, which is an enzyme. And it comes down and goes, and they keep moving through here. And then these float around and they hit another cloud of enzyme up here, which is polymerase. And it breaks open the the coding around the DNA and lets this come in and replace that segment. That's how you get immunities. All right, this is a little more geeky. These are all your different amino acids. And what is an amino acid? It's a very elegant little molecule. And these are all the different types. And you have 20 of them or so. And they come out in these long polypeptide chains from the bacteria. So the bacteria is over here going, well, actually it does it inside its body. And it gets it all figured out, and then it puts it in a ball like this inside its own body to create a ribosome. Later, when that goes out into the world, inside its own body, it's just going to create a ball. That's all. But it's going to be a program. It's all it is a program ball. When this ball goes out into the world and it bumps against something like this, all right, this ball comes out and everything's fine. It's floating around and it's going up to the brain, and some more goes over here and some more goes over here, and they're all locked in the ribosome inside the cytoplasm. But then somewhere way out over here, one of these nasty guys come in and it's attacking something. Well, what happens? A lysosome comes over and says, hey, you're the guy that takes care of those black things. There's one over here, he's wreaking havoc. This thing pops open into an enzyme, or he actually goes over there and probably hits it as the chemical signature and pops into an enzyme and just explodes it, kills it. That's what enzymes do, the ones that are the protector enzymes. And they float around in a fluid-filled highway, which drags them all around your body very, very quickly to attack invaders. Okay, I'm pretty sure these are the ribosomes, and they are squirting out these webbing of molecules that are coating all of the stuff that's coming in and going to kill it. That's what I believe is going on here. These are just arriving to unfurl and attack. I'm pretty sure. So don't forget, I explained how the ribosomes get up into past the blood-brain barrier, because it is permeable to very, very tiny things, which is the ribosomes, but it's impermeable to the proteins once they expand. So that's covered. All right, there's thousands and thousands of different species. Every species creates a different type of ribosome, if it's going to be one that creates ribosomes. I think some of them don't create ribosomes. They're consumer bacteria. But the ribosomes break things down. Some of them consume, some of them go out and spit out these ribosomes. And like I say, thousands and thousands and thousands, many thousands of these bacteria species. Each one of them is different. So inside of every cell, you're going to have a ton of those different little bacteria floating around. All right, they are in every cell. Now this is a little refresher just so you remember. Neurons, which is your brain cells, which are supposed to be restricted from all big chemicals, contain ribosomes. Now this is where AI is wrong again. They're saying ribosomes are large, which they are not, multi-protein organelles that synthesize proteins. The ribosomes do synthesize the proteins. They pop out as proteins, and they're in all living cells. So ribosomes are in all living cells. They just float around and they just collect in the cytoplasm. Now ribosomes are found in the neuronal cytoplasm. I showed you that before, I think, along with other organelles like the nucleus, mitochondria, and all that stuff, Golgi apparatus, yes. But they're not large multi-protein organelles. They are small ribosome balls. And they call them triggers. You can call them whatever you want, but they're ribosomes, little tiny balls. And that's the only way they can get through the blood-brain barrier. And they're little ones like this, and they pop right through, but when they get called on to do their job, they do this. Never going to get through again. Don't forget, bacteria are the factories that produce the enzymes. You have to have the bacteria. Here it is again. Bacteria, complex chemicals produced by bacteria, speed up chemical reactions without being used themselves. And they are not alive. They just are a chemistry boost. All right. Enzymes, you have to have them. And they only come from bacteria. And they're complex chemicals produced by bacteria. They speed up chemical reactions and they don't get used up. They're incredible. And they're only produced by bacteria. All right, you see this? This is about microbes that are on you. On and in you, your body full of microbes. You can't see them, but they're microbes. They're so tiny, you obviously can't see them. They're on you, they're in you, and you carry more than 100 trillion of them all by yourself. That's 14,000 times more than the number of people on Earth collectively. They're known as human microbiota. All right, these are the microorganisms. These are the bacteria. You and your 1.5 kilos of microbes. All right, and it gets in very deep, as I do too. A new view of the human body. They haven't thought about this in the right way. Mouthful of microbes, biggest organs, smallest protectors. This is the biggest organ now, is the interstitium, which they just realized recently. Slimy defense. Everything's about mucus. A hidden world in your intestines. Everything's about the bacteria created in the intestines. And from there, it appears to just shoot around. Intimate helpers. These are the little bacteria. They just do everything in your body. Pathogens, the bad guys. The expert explains. All right, so we're going to be going through all of that stuff. But this is what it is. It's the microbes. They literally control your body. They either make you sick or well, happy, sad. I mean, seriously. It's your emotions, your level of frustration and anger, sympathy, love, compassion. It's all part of that chemistry. Everything is controlled by chemistry. I don't care what anybody says. We are a chemical factory, and every one of those chemistry creates something. That's why you take drugs. You feel better. You don't feel hesitant. You feel that way. All kinds of things. You know, some things are very bad for you. It might make you feel well or better, but they're maybe not good for you. All right. Some things are good for you that you should have in your body and you don't have. So this is what it's all about. Your body full of microbes. So what am I asking for? This is what I'm asking for. All right, one last time and then I'm going to leave it at that. These are the ribosomes, the little blue balls, all in here. And they're not just going to be blue, they're going to be every different color because they're going to have some different effect. And the lysosomes are what calls on these things to do their chemistry. All the rest is just more things. All right, I'm going to leave it at this. The only way these ribosomes can get through the blood-brain barrier is to be as small as they are and not all puffed up with their opened up like this. Once they're like that, they can't get through. So it blocks those. Now, there's all kinds of ribosomes. Apparently, they just collect inside of every cell as just floating around in the cytoplasm. And I would assume there's all kinds of them in here, just waiting around. And when the lysosome pops them open, that's when they do their chemistry. And they do it like so fast, it's amazing. I think I showed you. It takes like something they were talking about. It takes 1 million years to do it if you didn't have the enzyme. Another one was like 400 years. And, you know, you have to have these enzymes. And how do they work so fast? They work in half-lives. You have to understand atomic theory, too, because you can go up on a periodic table and go down using enzymes to all of these. Iron, they can't account for anything above iron right now. in chemistry or in physics. Iron is where it stops. No. All of these are more particles bumped up. I call it atomic bump. Iron can go up to cobalt. Cobalt can go up to nickel. Nickel can go up to copper, zinc, gallium, all the way up to uranium, palladium. All of that stuff can bump up, not only down. They talk about decay mode. Well, I better show you. All right, get ready. You're going to be a nuclear physicist in a minute. This is iron. The next one up is cobalt. Well, what is cobalt? Well, cobalt, there's 28 different cobalts. Well, how could there be 28 of them? I thought cobalt was cobalt. It's not. There's 28 known, which is not even nearly close, radioisotopes, which means they can give off or gain energy, and one stable isotope. Cobalt 59 is the stable one. It occurs naturally because it has decayed to stability, given off or taken on, whatever it means to be not wanting or giving any more off or taking any on. That's what stability is. It's a range of stability. The radioisotope range in atomic weight is from 47 to 75. From 47 to 75. The most stable of them are 60. Cobalt 57, cobalt 56, and cobalt 58. The half-lives of these isotopes are, the half-life means they decay over a certain period of time. They got years, days. They can be instantaneous. But as we know, an enzyme can make something happen that would take 400 years happen in less than a second. So it doesn't matter what the half-lives are. They can bump this stuff up. And when I say bump it up, what do I mean by that? Well, cobalt is here. All right. Usually they say, well, cobalt is going to decay and go down. Okay, if you're not into physics or chemistry much, this will be a shocker to you, because everybody thinks each one of these atoms is stable, then the next one is just exactly that, the next one exactly that. It's not. There are all kinds of variations between those. They're called isotopes. And they have no clue how many isotopes there really is. This is isotopes of cobalt. Now don't forget, cobalt is above iron. All right? So cobalt can decay. All right, let's just take this one time. As you know, I assume things I shouldn't assume. Here's the headers here. Nuclide, which means how big it is, the heaviness of it. All right, let me open this up a little bit. All right, here we go. Now, the nuclide is how big it is. 47, 48, 49, 50. Why is that? 27 and 20 is 47. 27 and 21 is 48, 49, 50. And that's what happens. These right here, they consider to be the proton number, which designates what element it is, which is cobalt. So 27 protons means it's cobalt. But then they throw in all these neutrons. Well, what's a neutron? A neutron is only one electron bigger than a proton. So this is what I say a proton is. A proton is not this. There is no way that the standard model can work with this. It just doesn't work. The only way it can work is with the dipole electron flood theory, meaning that the nucleus of every proton is not made of big gigantic balls like this. It's made like this, where you can lose one little bit and piece at a time and drop down. See the actual weight of them. This is the mass. 47, 48, almost 49, 49, almost 50, 50, 51, and it goes all the way up the line. Now, what happens when these decay, which means they give off a couple of particles? All right, so now they don't have enough particles to keep them cobalt. They drop down. The daughter isotope is here. This is the decay mode. You don't have to worry about that. Just forget that. The half-life, this is how long they think they will last in this mode, is less than 35 nanoseconds, 44 milliseconds, microseconds, whatever it is. days, hours. Now, all of these are decaying from these cobalts and turning down into iron. So that's what you would expect. Well, it's going to decay and it's going to become iron. Well, guess what? As you go up, it can become nickel. You see that? They know this happens. It's stable here, but once you get above stability, you can turn it into nickel. And it's all over the place, nickel. You see this? Every one of them basically nickel. That's the decay is going up. That's not supposed to happen. All right, from cobalt it's going up to nickel. Well, can nickel go up to copper? I believe it can. All right, most people understand metals seem to be pretty conductive. Well, copper is among the highest conductor of all. So I'm going to be looking into this, but let me just talk about copper for 1 second. These are the isotopes of copper. Now, when they decay, copper can drop down to nickel or even cobalt. Okay, so it can drop way down or to nickel, which is normal. Now here, copper can go to zinc. Way down here, copper can decay to zinc. That's not supposed to happen until it hits stability. And where is stability? It's way, 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 way down here somewhere. All right, but see all this zinc? We can convert copper up to zinc. There's no problem at all. And zinc can go up to gallium. And gallium will go up to germanium. And right up the line. That's why they never understood this. But it takes enzymes to do this. You can't do this without enzymes. Enzymes break and make things in these half-lives. Well, here we come down to copper is stable. All right, at 65. All right. Even before we get to stability, we can get to zinc. It actually doesn't make sense to me, to be perfectly honest, because you zinc on both sides of stability. That's hard to understand, but copper is an exception to everything because it's so conductive. And I'm trying to figure out why it is so conductive. What makes that electricity flow through a copper wire really easy without getting resistance, getting hot, and just flowing through there. And the more, I would imagine, the more pure the copper, the better the conductivity. You see, this copper itself is the key. It says pure copper, known as electrolytic copper. Highest grade of copper has been the best conductivity. This pure copper is more than 99.9% pure and is made using electrolysis. Even the slightest decrease in purity can reduce its conductivity. So it is actually the copper itself. All right, so they're trying to address Alzheimer's, but I'm going to tell you something right now. All neurological disorders, in my opinion, other than damage caused by trauma, are linked. Now, this is Johns Hopkins on neurological disorders. I went to Johns Hopkins for genomics, which is basically the signals, really the programs. Genomics means there's a gene. There's a sequence of codes in your DNA that are a program, and they do a certain job, but you have to turn it on. If they never turn on, I don't care if the gene is perfect or not. If it's never turned on, it doesn't do the job. And what turns it on is called methylation. What is methylation? It means it's an enzyme that comes up and says, okay, we need you now. Click, and it goes out and it does its job. Otherwise, it just sits dormant, waiting. And what's its job? Its job is to turn on a ribosome. And the ribosome is the enzyme. It pops out like a gigantic flower and attacks or does whatever it's supposed to do. So you got the blood-brain barrier, which will not let these giant flowers through, but will let ribosomes through because they're tiny. And they just sit around waiting up in your neurons, in your cells. If they get a signal, they pop open. But you don't have the ribosome, it will never pop open. And you don't have the ribosomes, there's a couple of reasons. One of them is that you don't have the bacteria to make the ribosomes. That's the main part. That's what we're going to focus on, is having the bacteria to create the ribosome, which gets up through the blood-brain barrier. Then it's becoming already, it's an instrument of destruction to kill off or to create new proteins, which are the cell membranes. And that's what protects these signals. So just like, I'm going to be showing you this in great detail. Like I say, I don't go do things halfway. Now, this was Johns Hopkins. Here's 400 neurological disorders. 400. And here's all disorders. National Institute. Let's see what they have to say about this. Wow. These are all the different neurological disorders. Acid lipase disease, Alzheimer's disease, Angelman syndrome, whatever that is. And then look, all of these are different diseases. Now, are they, neuronal, blah, 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 neurological complications of HIV. This is all about the nerves and how they react to different invasions. And these are all invasions. Anytime your membranes get invaded, then you're going to be ill. And all of these things are nervous system, neurological disorders, and stroke. The stroke part is what intrigues me, because that just happens like that, and all of a sudden, and sometimes you recover. Now, why did you get the stroke? What was the cause of the stroke? And how do you recover? There was no damage, or was there some damage, or total damage? And why are those different situations? Because some people never recover. Some people recover pretty quick. A friend of mine had a stroke not too long ago, Pete, and he has seemed to recover well. He's quite satisfied with his recovery. Now, these are the things we're going to talk about. And again, it's going to be deep, so stick in. All right, so what am I asking for? I am asking for this: a fecal bacteria database. All that is a poop swab. So you take a spectral analysis of a fecal swab. Spectral analysis just tells you what's in there, and you'll be able to tell what kind of bacteria is in there, using AI to analyze the different light signatures. All right, like this is going to be a different bacteria than this. It's just as simple as that. Now, you're going to compare this signature that I have to the database, which is the fecal bacteria database, which is a broad array of a wide variety of people having all kinds of different symptoms. And sooner or later, they're going to fall into some form of a pattern. I'm almost 100% sure, because every symptom is going to be triggered by some kind of chemistry. So we want to be able to compare your gut fecal swab to the database. Then we're going to build a bacteria to conditions, bacteria versus conditions. And like I say, they're going to flow, and then they're going to show this. Everybody that's missing this bacteria is up here on some kind of disease scale, and everybody that has it is down here, that type of thing. Now, we want to create a study. So we need a study, and they're working on this right now in Northern Ireland. So we want to use their data and their expertise already. Marguerite O'Reilly, who I've been working with for 5 years or so, and they're going to need help. Nobody's got a lot of extra money to do this, but it's the effect on the healthcare system and the lives of the people with autism, particularly a young kid. Your whole life is going to be affected by that. And when I started with her, her son was nonverbal and had all kinds of issues, and they were going to put him in a locked ward. And after some modification of his gut stuff, he turned around pretty good. He's doing fine. And she has taken this into, really took it to heart. She went to Harvard to study the ribosome, all the things we've been talking about, Schwann cells. And I've been working with it, and she's been working with all kinds of people over there in Northern Ireland. Now, it requires probiotics at this point, and they're kind of expensive, and some other things. So we're trying to work on a little charity thing. and because these people don't have any hope, and their kids, it's just terrible. It's just heartbreaking to hear the stories, and then to hear the success stories. It's just mind-blowing that this is not taken more seriously. And like I say, I don't give any medical advice. This is all research, and Marguerite is doing the research, so I'll put a link to where she can be found. All right, I love you all, and everything leads to something else. So Alzheimer is leading to every other disease, really, and there's 400 types of cancers. And what does that mean? Every cancer is an invasion. All right, it's an invasion by your membrane. All right, here's cancer right here. It drills right down through, and if there's nobody in that fluid-filled highway to stop it, right inside, and then it grows these big tumors and causes all kinds of havoc. And then it can just split inside your body and spin around here and cause all kinds of terrible things. And here's the different stages. All right, distant spread. Once it gets through the regional spread, that means it popped through that lower layer, and then once it's in there, these invaders can move around and take advantage of other susceptible areas. Because if you're susceptible in one area, you're going to be susceptible in other areas. It's because the bacteria have to be everywhere. And they seem to go everywhere, apparently, in those ribosomes areas, just sitting around waiting to be used. That's what it appears to me.